So the concept of leukemic stem cells was introduced in 1997 in a seminal study published in Nature Medicine by Dominique Bonnet and John Dick. This study was cited more than 4,000 times. So Dominique took leukemic cells from AML patients and sorted for phenotypic stem cells, that is CD34 positive and CD38 negative cells, and more mature progenitor cells, that is double positive cells for CD34 and 38. Only the immature cells produced leukemias, but not the mature cells. So they propose that stem cells are the quiescent are the disease initiating cells. While this is true for a certain group of patients in the study, it's not the case for all leukemia subtypes. So leukemic stem cells, like normal stem cells, can self-renew and differentiate to blast cells, which are the major leukemic cell populations. However, these blast cells cannot differentiate to mature granulocytes or monocytes. But these LSCs can come from both hematopoietic stem cells as well as mature progenitor cells like CMPs, GMPs, or MEPs. You also have to consider stepwise addition of mutations for leukemogenesis. The pre-leukemic mutations may have been acquired at stem cell stage, but the secondary events can occur at a progenitor level, and then that can cause leukemia. So these leukemic cells are often resistant to standard chemotherapy, for example, cytarabine or daunorubicin or hydroxyurea. Of course, the major blast cells would be eliminated, leaving the stem cells to contribute to disease relapse, disease recurrence. So therefore, it's important to eliminate both leukemic stem cells as well as blast cells. So what are the factors that one needs to look when considering targeting leukemic stem cells? Obviously, the first thing is specificity or selectivity for leukemic stem cells. The second important thing is that the drug should have minimal effect on normal stem cells. Okay? Otherwise, the patients will have bone marrow failure. Third aspect to consider is heterogeneity within leukemic stem cells. Okay, so there will be multiple subclones of leukemic stem cells, and you should be able to target all of them. So how do we go about looking for a drug? Okay, we can look at the following aspects. We can see how leukemic stem cells are different in terms of their immunophenotype, metabolism, transcriptome, epigenome, and their spatial localization. So I'm going to discuss how we could use this information by taking specific examples. So each of these antigens listed here are upregulated in leukemic stem cells, at least some leukemic stem cells from some patients. So there's no one antigen that is expressed on all leukemic stem cells, unfortunately. So one approach is using antibodies against this antigen and eliminate all or some of these leukemic stem cells. These antibodies can induce antibody-dependent cytotoxicity. We can also use a bi or tri-specific antibodies by redirecting T or NK cells. So the other approach is linking antibodies to various toxins to create a targeted delivery vehicle. In our lab, we're actually using this approach. We've developed a novel peptide drug conjugates wherein these peptides specifically bind to some of the leukemic antigens and act in the delivery of cytotoxic payloads into cells. Okay, we can also use this information to target LSC-specific we can also use this information of these LSC-specific antigens to understand the biology of the leukemic stem cells. Here I'm talking about an exemplary LSC antigen called CD123. CD123 is highly expressed in LSCs from high-risk MDS patients, and it's not expressed on low-risk MDS patients, as you can see here. So in this Nature Communication paper, what they have done is they've sorted for CD123 positive cells and CD123 negative cells and performed their transcriptome. So they found that translation and inflammation are highly upregulated in CD123 positive cells. So they verified this using looking at the translational rate in CD123 positive cells when it's compared to CD123 negative cells. And then they used omacitaxine, a translation inhibitor, and they found that this inhibitor specifically targets CD123 positive cells shown in blue compared to CD123 negative cells. Okay. The next, the targeting. Next, they used this inhibitor in mice. So they injected MDS cells into mice and treated the mice after successful engraftment with omacitaxine alone, or in combination with the standard drugs like azacitidine, or which is a DNMT inhibitor, or venetoclax, which is a BCL2 inhibitor. As you can see on the right, and they found a greater reduction in the number of malignant stem cells either alone or in combination. Okay, interestingly, this drug does not affect the normal stem cells, as you can see here, and this compound acts by inhibiting the translation as shown here. Okay, so next, targeting LSCs based on the phenotype. Okay, that's what we're going to discuss. It's difficult to target transcription factors that are aberrantly active, but the epigenetic modifiers the transcription factors rely on can be easily targeted. Okay, so here on the left, some of the factors whose modulators are involved in a variety of clinical trials. Okay, so this particular study was led by Professor Tim Somerville from our university. They chose to target LSD1, which is histone demethylase, because LSD1 levels are linked to colony forming capacity, which is a kind of a surrogate marker for leukemic stem cells. So they've silenced LSD1 using a shRNA in AML cell lines, okay, as you see here, and the silencing of effectively reduce the CFU capacity. Then the drug discovery unit from Manchester again developed two inhibitors, and the compound B, as you see here, it can reduce a clonogenic capacity at a lower concentration. Okay. And then they wanted to check this compound in vivo. So they transplanted AML cells into mice and treated them with compound B. As you see below, the number of leukemic cells from these mice have reduced compared to the controls, okay? And moreover, stem cells recovered from these mice have reduced CFU capacity. Moving on to targeting altered metabolism. LSCs are often thought to be quiescent, but it's not always the case. Some LSCs are also proliferative. So LSCs differ from their normal counterpart in terms of mitochondrial metabolism, inflammatory pathways, glutathione balance. Again, there are several inhibitors targeting this altered metabolism are in clinical trials. I'm going to focus on inflammation. Okay. In this study, they looked at NF-κB activity, that is inflammation, in control, and MPN, which is the myeloproliferative neoplasm mouse models. Okay, this NF-κB activity is measured by the luciferase activity as shown here, and you see MPN mice have got increase in inflammation compared to controls. So they tried to target this inflammation using a small molecule modulator called JQ1. JQ1 targets the bromodamine containing protein called BRD4, and thereby it inhibits NF-κB targets. As you see here, treating MPN models with this JQ1 reduces some of the inflammatory cytokine productions. Okay, this further contributed to overall survival of these mice as well. Okay. Moving on, leukemic stem cells also hijack bone marrow microenvironment for their own survival. The bone marrow microenvironment is composed of multiple different cell types, as you may know, mesenchymal stem cells, sinusoidal endothelial cells, osteoblasts and osteoclasts, and then also non-living extracellular matrix like osteopontin, fibronectin, hyaluronan. Okay, all of these factors facilitate the addition of leukemic stem cells and AML cells to the bone marrow niche. So several interactions are depicted as you can see here. Okay, and these interactions protect these LSCs from chemotherapy. And all of these interactions are under clinical investigation for therapeutic benefit. Now, let's discuss some of the latest developments in targeting leukemic stem cells. So, in an attempt to differentiate leukemic stem cells, authors from Oxford have used LSD1 inhibitor along with a GSK3 inhibitor, which induce differentiation of LSCs but not affected healthy stem cells. In another study published in Nature Communication paper, authors have used bispecific antibodies targeting LSC antigen CD123 and CD16, which enhanced NK cell function. Menin inhibitors could be used to target LSCs because it reduces LSC gene expression program, thereby could eliminate leukemic stem cells. There are a number of menin inhibitors that are currently FDA approved or in clinical trials. Okay, so ultimately, effective elimination of LSCs requires combination therapy.